So in this next video, I'm going to be filling you in on how a neuron integrates information that it receives from other neurons. Through dendritic spines, a neuron can establish more than 50,000 connections to other neurons. Nerve impulses traveling from other neurons bombard the receiving neuron with all manner of inputs, excitatory and inhibitory. So what this means is that if you were to picture a neuron as a bit of like a tree, okay, and you have the main trunk of the tree, which would be the axon, and then you would have all the dendritic spines and branches, and those would be the branches of the tree. So the trunk can get information from all of these branches. So you can have a really intricate, interconnected branch system that will send information down through the trunk. And that's very much how some neurons can look, with a sort of intricate dendrite branch system, and all of those dendrites have the potential to establish with other neurons. The cell body, which is located between the dendritic tree and the axon, can receive inputs from other neurons as well. So it's not just incoming from the branches, you can have the cell body. The inputs that are received from these other neurons can be either excitatory or inhibitory. So what do you think an excitatory input would do? And this is obviously a theoretical question because we're having online video. But think about it for a moment and think about the word, okay, excitation, excite, if you are all fired up, to use a little bit of an example here. So excitation would encourage a neuron to fire. Inhibitory signals, on the other hand, if you think of the word inhibitory, you think of, okay, someone's going to inhibit you from doing something, then they're going to stop you. So inhibitory signals are going to try to stop the neuron from firing. So how does the neuron integrate all of the excitatory and inhibitory signals that come into a nerve impulse? Well, first, let's go over a little bit of nomenclature. Excitatory input is known as excitatory postsynaptic potential. Okay, so this is a brief depolarization or increase in positivity of the neuron membrane in response to stimulation that makes the neuron more likely to produce an action potential. Inhibitory postsynaptic potential, on the other hand, known as an IPSP, this is a brief hyperpolarization, making the cell more negative, okay, in response to stimulation. This makes the neuron less likely to produce an action potential. Now, the EPSP and the IPSP, those abbreviations are going to be used throughout the course. So you'll actually really want to become familiar with that abbreviation. It's one of those things that if you don't catch that or keep careful note of that abbreviation, that it can be confusing if you were to encounter it in the subsequent chapters and not remember what it means. So an excitatory postsynaptic potential, an EPSP, it's going to encourage the cell to fire. And an inhibitory postsynaptic potential, or an IPSP, is going to stop the cell from firing, or it's going to try to stop the cell from firing. So the EPSPs and the IPSPs that are received from the neuron are summed. They are summed in two ways. You have temporal summation and you have spatial summation. Temporal summation are pulses that occur at approximately the same time on the membrane. And so those would be from separate neurons, an EPA from one neuron and an EPA from another neuron, but they occur at relatively the same time, so they can have an additive effect on the receiving neuron. Spatial summation, however, has to do with location. So pulses that occur at approximately the same location on the membrane are also summed. So it would be like you have your tree with its trunk and then have all the dendritic branches. If you have two EPSPs that are received on one particular branch as opposed to on opposite ends of the tree, if they're received on the same branch or in the same basic location or vicinity, then their information will also be able to be summed. Here's an example of temporal summation. So if you have a wide temporal spacing between EPSPs, then they're not going to sum. The whole goal is to reach threshold. The reason that anything is summed, particularly EPSPs, the only reason they sum is that one EPSP is not sufficient for a neuron to reach threshold. So you're going to have more than one. And this is something that would be really important for if you just had one EPSP that was able to be, that would say like, okay, to pass another neuron to fire, then your brain would almost be too noisy. So a signal coming in from other neurons has to be strong enough to reach threshold, and only information that is strong enough would cause a neuron to fire. So if you have wide temporal spacing, then you don't have any of the summation that would be required to reach threshold. If, however, they are close in time, then you have a much higher chance of reaching threshold. So if you have stimulus 1, stimulus 2, then they build on each other, and after stimulus 1, then stimulus 2 will be higher. Similarly, if they occur almost at nearly identical times, then they can sum to result in a larger graded potential that brings the neuron even closer to threshold for firing. IPSPs that try to reduce the firing of a neuron operate on a similar principle. If they occur farther apart, then the impact on trying to prevent that neuron from firing is less strong. If they occur in close temporal proximity, then their impact on trying to prevent the neuron from firing is stronger. Here's an example of spatial summation. So here, again, I gave this example across the tree here. You have these opposite sides. If you have them come on opposite sides, they could be, even if they're at the same time, but they're on separate parts of the membrane, then they're not going to influence each other. EPSPs produced at the same time, however, and close together can form a larger EPSP, a larger potential that passes threshold and will enable the neuron to fire. If the summed EPSPs and IPSPs on the dendritic tree in the cell body charge the membrane, change, this should be change. So if the summed EPSPs and IPSPs on the dendritic tree and the cell body change the membrane threshold at the axon hillock, which is right here, and this is really important. So it's not that you can just, oh, you have an EPSP that comes in up here. The EPSPs or the IPSPs, but primarily only if the EPSPs change the membrane potential at this threshold, only then will an action potential be generated. And then after it's generated, it will travel down here. So if you have EPSPs that are coming in here, but their effect is not strong enough to change the potential down here, then even if you're getting lots of information up here, if they're not additive, the signal isn't strong enough, then it's not going to cause an action potential. Now I'm going to take a second to draw some of this out and show you how this would look in greater detail. Okay, so now I'm going to hopefully provide you some additional explanation with how EPSPs and IPSPs are summed across the dendritic trees in the cell body of a neuron. So here is our rudimentary neuron, and I hope you guys can see this well enough. Try to put another color. Oh, I think blue is going to work best. Okay, so here is your axon hillock. This is the point, okay? And this is your cell body, and these are just some dendritic spines, make up your dendritic tree, all these branches here. So when an EPSP comes in, okay, if you have an EPSP that comes in here, okay, it has a lot farther to get down here. So if this has to travel all the way down here, then this is going to have to fire repeatedly to cause the graded potential. And again, this graded potential is just is basically a membrane change. Okay, so for an EPSP, it's positive. It's sort of a positive change. For IPSP, it's more negative. So this increase in positivity is going to need to come down here, and it's going to need to come all the way down here. So this section is going to have to receive so much input, then the positivity, the sodium ions, continue to sort of flow in all the way down here to the axon hillock. Okay, and we'll make these green. These EPSPs green, so it's like, okay, greens go for fire. So all of these, the graded potential has to travel all the way down here to get to the axon hillock. And then if it gets down here, then it will fire, and it will travel all the way down to the terminal buttons at the end of the axon. So typically, a cell is going to be getting multiple EPSPs at once. So you can have EPSPs that be coming in from here, okay? You could have some coming in from here. You could have some coming in from here. At the same time, they're also going to be getting some IPSPs, some inhibitory signals, and we'll make these red. Stands for stop. Oops, these should be minuses. So these are these sort of stop signals that are coming in. So you can have your IPSPs that are also coming in at various sections throughout the neuron, okay? This is making the potential, this area more positive. This is making this area more negative. All of this, the whole point of it is to result in an action potential here at particular times, okay? Neurons, you know, they're going to fire sometimes, so it's just a matter of having them fire at the opportune times. You don't ever, if a neuron doesn't ever fire, then it's actually not being used, and it will die out over time. This is part of what occurs in development. It's called neural pruning. Okay, it particularly occurs early in development. So it's just an interplay of like, okay, other cells, other neurons telling this neuron to fire, and other ones saying, no, 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 not quite yet, not the right time. Okay. So you have these positive things coming in. If this graded potential reaches here, an action potential begins. And so this is where this sort of summing occurs. If you get lots of stuff here, then even if you get some IPSPs that are coming in, see at the opposite side, remember the sort of the spatial summation, if they're coming in at opposite sides, then there's really not a chance for them to group together and result in a greater membrane change for more positivity. If, however, they're coming in at a similar location, okay, then you have a chance here for these two to join together and make this even more positive and result in an action potential. And the same thing goes for negative, okay? If these were here to come in and make stuff even more negative here, this EPSPs would have a greater potential of stopping an action potential, of preventing the positivity that would result in an action potential. So EPSPs are best summed when they occur at a similar time, the signals come in here at a similar time, and space, then they have the largest impact. So a question that I've gotten in a previous class, someone actually asked if there's a particular location, so say that if there's EPSPs that come down here, if this is more associated with epilepsy. This is not the case, but it does bring up an excellent point. So where the IPSPs or EPSPs come in from really largely has to do with the connections with other neurons. But there are a couple of different important things to realize, is that again, the EPSPs that come up here, they have lots of space to travel from. These, they don't have as much travel to do. So these, the connections with these other neurons, these neurons will have a much greater chance of causing this neuron to fire, these connections. So they have a stronger relationship. So what generally happens with neurons is if they fire together, they wire together. So if one causes the other to fire and they happen to fire in sequence, then those connections are actually going to become stronger. And they can change a little bit. The connections, these dendritic spine and tree connections are malleable. Major structural differences, it's not something that they move around a lot and like, oh, the branches are waving in the wind or something like that. No, they are a little bit more structural than that. But they do go through subtle changes that can increase the likelihood. So you could, for example, find that this neuron, the neuron that comes over here, this is often stimulating this. And so it's going then, neuron that comes in over here, this is another neuron, it's going to come in and cause the EPSPs here that travel down. Well, it could over time try to form another connection to even strengthen the connection here, so it can have two axon terminals that end here and increase the likelihood of having some potential right here. So it is possible for neurons that are used heavily to become even more strongly connected, so they can basically be more synergistic. If I fire, then you fire. This could be advantageous in some ways. Now, you can also see how habits tend to develop if you tend to do one thing that's associated with something else, even at a neural level. So again, if you fire together, if you wire together, you fire together. Now, the placement of these is also opportune with regards to preventing behavior. Okay, so say that you want to try to prevent smoking or some other habit. You're going to have to then when you try to change that habit and the actions that you do, you're actually trying to make structural changes in the brain that reflect the ceasing of that particular type of behavior. So if smoking, was associated with something that you typically did in certain social situations, when you went out to drink, and or when you were hanging out with particular types of friends, then seeing those friends would be a cue, or even going into having a drink at a bar could be a cue for engaging in smoking behavior. That these would sort of help and cause this sort of behavior to fire. And again, this is, I'm sort of talking about a large-scale relationship that it's not like one neuron is associated with a bar and another neuron is smoking with smoking behavior. But I'm illustrating a larger point in terms of the connectivity between stimuli and firing and the implementation of certain behaviors. If you are trying to change a behavior, then it is going to need to occur at a neural level. And you'll need to have the formation of new connections that come in. And so if, say, you're trying to quit smoking, and you're doing a really good job, and you're trying to engage in new behaviors, and so then you'll have additional neurons that can come in here and stimulate these areas that would result in a greater chance of preventing this neuron from firing or preventing from engaging in smoking behavior. So you can take this example and apply it to a range of behavioral connections, but basically neurons and the pattern with which they fire and even aspects of their structural relationship, where EPSPs come in, where IPSPs come in, that's going to reflect aspects of your behavior, what you learn, what you know. Anytime you're learning something new, there's going to be really small structural changes that are going to that are going to represent the addition of that knowledge to your neural network. And so those small changes over time, and you can see from EPSPs here, or adding some other ones up here, or something like that, can have these small changes. And again, it can take a long time. It takes a long time for you to learn information really well. But once you do, there's going to be structural changes that are going to reflect the depth of your knowledge. And once you do know something with regards to a particular field, what you'll find, and this is reflected at the neural level, that it's easier to learn newly related information because you have an existing framework with which to add this new information to. So this is in general a little bit more about how EPSPs and IPSPs work to increase or decrease the firing rate of a neuron. One other thing that you'll want to know is that you have a neuron, or the relationship between two neurons, is either inhibitory or excitatory, okay? So if I have one neuron that's coming in here, okay, and I have another neuron that's coming in here, then this, say this neuron and this, it has an EPSP, and this one has an IPSP. That means that this neuron, it will always result in EPSP. So if these two neurons have an inhibitory relationship, this neuron is always trying to inhibit this one. These two neurons have an excitatory relationship. This one is always trying to excite this one. Okay? So it can't change. So this relationship is inhibitory, this relationship is excitatory. It's not like, oh, this relationship is excitatory, but now this neuron is going to suddenly change and it's going to be in IPSP. That doesn't work, okay? So that's not how it works. The relationships are set in terms of this neuron always has this type of relationship on the cell, this one always has this type of relationship on the cell, and so on and so forth. And that is the basics for EPSP and IPSPs. So that's the basics for how they're going to sum across time. And it's really important, the relationship. So again, the relationships here, that the incoming information that's right near the axon hillock, this can have a much greater impact on whether the neuron fires or not than those coming here. And I guess that's it for now.